I was eating these things over the coffee break, and they are brown and full of ages and glycation end products. Don't eat them, they're really bad for you. Okay, we've talked a lot about what are ages already, so I'm going to skip quite a lot of this. A little bit about what they do to you, but primarily focus on how you might remove them to come to precisely that question. Warning, there's chemistry here. So, the age production pathway we've talked about, so I'm going to skip this really quickly. That happens quickly in chickens, roast chickens obviously. That happens, that happens somewhat more slowly in us, but it does accumulate. I'm not going to go to that. This is a little bit more detail on the chemistry. And I want to point out the key role of dicarbonyls in the centre here. People have mentioned them already, both as oxidation products of the amidori products, as oxidation products of sugars, and as products of the lipid oxidation pathway down there. We're going to come back to dicarbonyls in a minute. The key thing to remember about ages is that people talk about age as if it's one thing, the advanced glycation end products. This is a zoo of materials in here. Some of them are coloured, which is why the lens on the previous slide was a sort of yellow colour. Some of them are fluorescent. Most of them are chemically stable, so they hang around for a long time. But to give some idea of the sort of zoo of products, here's a partial list of those that are known to accumulate in vivo. You can make others in the test tube. And the thing to remember here is that, so I've got the amino acid bits in red, they've reacted with the sugar bits in black, and they fall into two types of classes. Ones in which you have an amino acid modified by a bit of sugar carbon, which is called monovalent, so this is altering the structure of the amino acid. And others in which you've got two amino acid bits linked by carbon coming from sugar moiety. And those are divalent, they cross-link proteins. So we've heard about protein cross-linking before. And so their effects they might have are obviously to cross-link protein, mechanically constrain them, but also to modify their properties. Through activating RAGE, people mentioned RAGE, degrading protein structure and function, as Andreas mentioned before with PGSF, or to generate reactive oxygen species. So there's a soup of material here, there's a zoo of possible properties. And I use that terminology particularly. The most common cross-link is probably this one, glucosapane. The most numerically common, and probably not the most physiologically relevant, is this one, carboxymethyl lysine. But I'm not going to talk a lot about the chemistry of those because that gets fairly dull. A short digression on how you measure them. We've heard about fluorescence. That's a great way of measuring some sort of total aggregate amount of glycation going on, but it doesn't tell you what ages are there. It doesn't tell you where they are, and it doesn't tell you, except semi-quantitatively, how much there are. There's a conventional method, HPLC-MS, conventional chemistry lab stuff, very slow, very expensive, very skill intensive. There are only a few groups in the world that do that. There are much faster immunoassay methods, and my bottom line from those is that they are all poo. Okay? And they're all poo for this reason. We did a long study on this, looking at anti-age antibodies and another cross-link antibody that I'll come on to in a minute, and looked at what things they react with. And almost all the antibodies react with things other than ages. Some of them don't react with ages at all. They react with serum albumin. So if you see a paper that says we measured this particular age using an antibody, don't believe it unless you've personally looked at that antibody and checked it actually works. A lot of these studies looking at antibodies, looking at ages, essentially use chicken soup as a reagent. Okay, so they take protein, they take sugar, they cook it up for 6 to 8 weeks at 37 degrees. You get this brown goo. It's chicken soup. It is not a chemically defined material. They do not know what they're looking at. Stick to HPLC-based methods. Okay, that's all I'm going to talk about what are ages. What do they do to you? And there are two ways of looking at what ages do to you. You can try to work out the mechanism of what they're doing in particular cellular systems, or you can say, we don't know, let's take them out and see what happens. Okay, classic biology way of doing things. I'm going to knock something out and see what happens. Physicists think this is a very strange way of thinking. So this is correlation of ages with disease and with age itself. Different chemical ages along the top here, different tissues down here. People have looked at the lens of the eye a lot because it's easier to get. They looked at the skin because you can actually get at it. Taking brain biopsies is quite tricky on living people. Taking skin biopsies is easy. And of course it's serum. These are all HPLC-MS studies, so they actually are measuring the ages. Fluorescence is also a really good disease marker, okay, but we'll come on to marker in a minute. And blue means it's correlated with age, red means it's correlated with disease, primarily diabetic complications, and the other colours mean it isn't. And there's an overwhelming consensus that these things build up with age, and they are correlated with disease. So, okay, that would suggest ages are bad. The problem is this, correlation or causation. If I did a survey on this room and asked what decade your favourite pop group was most active in, so decade along the bottom here, I guarantee that would correlate very well negatively with age. Okay, so, you know, you're around my sort of age, you like Stoned and Status Quo and the like. Half of you like Martin Garrix and the like. But the problem is, if you made me listen to Martin Garrix, you know, that wouldn't happen. There he is, horrible young man, revolting music. The point is, this is a correlation, it's not a causation. And so the question with age is, are these things just accumulating because of something else happening, or are they causal? Right. So, there's lots of evidence that specific pathological mechanisms are correlated with the accumulation of age. So this is talking about cross-linking. I'm going to whiz through this very quickly because we had a great lecture on this just before coffee. This is another bit of blood vessel. I don't chop up people. This is from somebody else who chops up people. Aortic stiffening, we talked about, retinal degeneration, tendon, bone, all these things. We lose elasticity, we lose the ability of tissues to be remoulded, and as we heard in the last talk, we also have the extracellular matrix of old people. Cross-link matrix has less ability to support stem cells, support cell migration, and so on. The problem with these sort of studies is that we don't know what sort of cross-link is relevant. Sorry, we don't. We can hypothesize its age, but it's not necessarily. This is another sort of cross-link. This is a cross-link formed between a lysine and a glutamine by the enzyme transglutaminase. We all have this. It's greatly upregulated during chronic inflammation. If you go out to a restaurant there and eat cheap food, it's very difficult to do in Brussels, but if you do, and you get a piece of meat that looks like this, it's got this sort of uniform rubbery texture to it, and you bounce your fork and it sort of goes boing rather than squidge. The chances are it's cheap meat that's been cross-linked by transglutaminase. This is widely used in the food industry. And this is the distribution of glucosapine, the most common cross-link in skin. Blue in non-diabetics, red in diabetics. This is concentration up here. And on the same scale here, I have 3 studies looking at this transglutaminase cross-link in healthy and in a couple of disease states. Now the problem is nobody in the entire planet, nobody has measured both of these at the same time, so it's rather difficult to say whether one's more common than the other in the same tissue, in the same person. But matching up these 2 different studies like this, it suggests that this cross-link is at least as common in disease states as the most common age cross-link in the most diseased and sick diabetics. So we don't know whether age cross-links is genuinely important. We can hypothesize, but we do not know. Receptor for advanced glycation end-products is another route. At least 4 different age-binding proteins. This is the most commonly studied. Found in lots of sites, particularly in the lung. A very promiscuous binding protein. It binds to all these things, several proteins, different sorts of ages and things. BSA age here. This is the common reagent. This is the chicken soup. All right? These people are measuring the binding of this receptor to chicken soup, not to a defined chemical entity. And if you ask yourself what is the affinity of this, and then for these different ligands, how much of them are present in the body, not how much are present in the test tube where you tested this, but in the actual body, you find that for most of these ligands, I've actually started doing this, this is not complete yet, this is the fraction saturation of the receptor. In other words, how much of that ligand will bind to the receptor, given what's going on in the body. For nearly all of them, it's almost nothing. So people say rage is stimulated by, and what they mean is we take a big tube of my favourite molecule and we drop it on rage and say, look, something's happened. That is not the same thing as what goes on in physiology. So, what is the role of age? There's 2 lines of evidence that suggest that advanced glycation end products do very little. The first is this one. I zipped through this side, I pointed out dicarbonyls, but there's this route here. There's an enzyme inside the cells that reverses the formation of amidori products. This is fructosamine 6 kinase, and people have made a fructosamine 6 kinase knockout mouse. They've knocked out that enzyme in the mouse, and the mouse is entirely normal. It has no obvious pathology at all. And when people do knockouts, they tend to say, okay, we looked at it for a week or two and it's fine, it's not dead. This was published in 2006. There's been nothing on it since, which suggests people have been looking at this thing saying, when's it going to get sick, when's it going to get sick? And it hasn't got sick. Okay, this is several mouse generations. This is only intracellular, so this is not affecting extracellular matrix glycation, probably. However, the other thing is this really interesting work from David Spiegel's lab in Yale that suggests that one of the glyoxyl-derived monovalent ages, MGH3, it actually has an antioxidant effect in vivo and might actually be protective against reactive oxygen species. So, I go through these to illustrate an important point. We do not know what ages do in vivo. We can speculate, we can hypothesize, but we don't know. So, what we're going to do is we're going to get rid of them. There are two ways of getting rid of age. One is to block their formation, the other is to say they've already formed, can we clear them out? And people have tried both. Age blockers, some more chemistry. Here's a partial list of things people have tried to block as potential blockers of age. Aminoguanidine, this small molecule up here, is a very commonly tested one. But there's a whole slew more, including dietary supplements, drugs, anything, any list of things that do good things for the diseases of aging. They include drugs, has to include aspirin. And I'm pleased to see that aspirin's in there. The clue for what's going on here is this one down here, deferoxamine, this thing. Looks like some sort of train line. It's actually an iron chelator, and it is used clinically to remove iron overload in thalassemia patients and the like. It is a very, very potent metal chelator. A number of these things are actually metal chelators. They don't directly interact with that glycation chemistry. What they're doing is removing metals and stopping oxidation going on. A number of others are antioxidants, and so clearly if you've got a process that's driven by oxidative damage, they're going to reduce that process. Not necessarily blocking ages directly, but they are antioxidant materials. People have put a lot of these things into animal models. They put a few of them into man, aminoguanidine primarily. Some improvement from an early study. A much larger later study was stopped later on because it seemed to be doing more harm than good. Benfotiamine is actually a pyridoxamine a precursor. Seemed to show some neuropathic symptoms in diabetics. Diabetics are notoriously prone to pyridoxamine deficiency. So you give them a pyridoxamine or pyridoxamine precursor and they got better. That's not saying anything about age. That's just saying about the metabolic deficiencies. And this experimental compound was pulled out. So the problem with the age blockers is not that you can't find something that does something, but deconvoluting that mechanism of action, working out what they are doing in people, at the moment is almost impossibly difficult. The most exciting approach is age breakers. Can you find a molecule that actually goes in and breaks those ages, and particularly the cross-links? Two have been tested out in man. Allegobrium, ALT711, this molecule here, and TRC4186 from Torrent, this molecule here. This was a sort of quasi-designed molecule. This was a result of screening. Both the groups researching these both so they break ages in vitro. The problem with that, and we can talk about why later on if we have time, is that the standard screen, how they describe, how they measure whether these things break ages in vitro, do not discover age breakers. They do not measure age breaking in vitro. I don't know what they do measure, but again, it's chicken soup chemistry. And it almost certainly not look at age breakers. What they probably do is discover chelators, detergents, all sorts of things. So, LT711 is the one that's been researched most. This is a very partial list of some animal studies. These are the rat studies are nearly all in diabetic rats. It looked at APOE knockout mouse, diabetic mice, and a couple in dogs and one in monkeys, and they say that this improves all these various parameters. Different parameters in different studies. Some studies say, for example, it helps renal biochemistry, other studies say no, it doesn't, but it does something else. It's a bit of a mixed message. Incidentally, in the last year, Dave Spiegel's group at Yale again have found some very interesting chemistry going on around ALT711, which suggests it does react very potently and specifically with dicarbonyls. But the very few of the cross-links in vivo are dicarbonyls. That's the intermediate in the middle of the slide, not the end product. In man, half a dozen studies in man, and there's the classic progression we see again and again and again with exciting new therapeutic approaches. An initial trial that says, yes, we've got some interesting looking sort of things, and then a follow-up trial that says, yes, we have, but they look really illogical, and then people follow up with more detail and cannot reproduce it, and the effect goes away. So in clinical trials, ALT711 has done zip. Sorry. So it does something in animals, mostly acutely diabetic animals. It probably does nothing in man, but the only thing it's been tested in is hypertensive older people, specifically that are not diabetic. So they tested young diabetic rats and old diabetic people, and are surprised, non-diabetic people are surprised when it doesn't do the same thing in both. I mean, it's like sort of, putting water into your horse and saying it walks, and then water into your car and saying it doesn't. I mean, it doesn't make sense. So this is not age breaking, it might be dicarbonyl scavenging. Compound, not at all clear what this does. This is the core part of the molecule that does whatever it does according to their patent, and I don't know what that does. There's some interesting chemistry from 20 years ago suggesting that this might actually be a dicarbonyl locker. It reacts with dicarbonyls and locks them up into a stable structure. That's the reverse of an age breaker, but if it's a dicarbonyl scavenging molecule, clearing that stuff out in circulation, that will work. And really interesting, the C-glucosyltryptophan, looking at the structure, it just might do the same sort of chemistry as this. I'm not sure, I need to go back and look at that, but it just might. So there might be some relationship between that really interesting marker of aging and glycation and glyoxal products. However, it's all chemistry stuff. Again, TRC4186 does interesting stuff in diabetic rats. It's gone into phase 1 in man, it's gone into phase 2 in man. The protocol was signed off at the end of 2009. It was last updated in 2013. We've had no results coming out from that in public at all, but the Torrent website still lists this as an active program. So they still think this does something. They don't know what. They are testing this in diabetics. So if it does do something in carbonyl scavenging, then that's what it does. Okay, so ages. Should we prevent them, remove them, or ignore them? Prevent them, yeah, but it's not practical. We do not have any way of probing that system to say if we block the formation of advanced glycation end products, what happens? Because all the molecules suggested as age blockers also do at least one other thing, and most of them mostly do one other thing. Oh, one of the other reasons the ALT711 trials are all deeply flawed is that on hypertensive people, those people are taking antihypertensive drugs. A number of those drugs are listed as potential age blockers because they are chelating agents and or free radical scavengers and or antioxidants. So they are modulating the amount of ages they have anyway. I mean, it's a mess. It really is. That's the trouble with doing medicine, all right? I mean, people are so messy. If only we could have a nice, well-controlled, inbred population of people that's not given any drugs and given Purina rat chow for 30 years, we'd be so much better off. So, prevention. Removal. Not yet doable. There are no age breakers yet known that work as age breakers. They might do something, but we don't know what, and they're not age breakers. I don't think ignoring them is wise. Clearly, the dicarbonyls, glyoxal and methylglyoxal are likely to be bad. They're reactive molecules. No, please don't eat this type of molecules. But we don't know how best to address them. And cross-linking is likely to be bad, but we do not know what cross-link we should be going after. And this is something that the research group in Cambridge was attempting to have a go at. But we are starting at last to get new tools to address this. And so that's what I want to thank on the last slide. We set up a lab in Cambridge a few years ago. Here they are. IRNR, your photo up there, less extreme hair then. Funded by the Centre Research Group, Aubrey of course, Michael's input a lot into this, and Jason funded the group. This work's now being transferred mostly to Jonathan Clark at Babraham, who doesn't look very pleased about it. And the chemistry at David's lab in Yale is trying to define exactly how you make and then how you test and then what the properties are of each individual glycation cross product. And this is Christian here holding in his hand a sample of pure glucosapane that he's made in the lab. He's very proud about that. So we are starting to get the tools from which we can answer at least some of the initial questions about what we should do about glycation end products. Thank you very much.
And so in my talk tomorrow I will go into the chemical details of the speaker lab. Any questions? No? Okay then. Oh, okay.
Do we even believe that we can see an effect of an HE break or something like this? So I just do not want to answer to Graham's question because I don't believe that anything works at the moment. In theory it may work, but if you, for example, look to the human aorta and you really go to an old patient when you want to treat him, to help him, he, for example, accumulates up to 1 pound of calcium in this aorta. You have a total mass amount of calcium accumulate, and then you modify a little bit the HE crosslinks. It's really hard to believe that you see anything, even if it works. If you look at the function of the aorta, for example. So you have a multifactorial system, and HEs are one part of it, and you modify one and not the other ones. So it is really, really difficult to believe that even if it works, we can see anything in the function.
Yeah, and this is, there's a long answer to that. The short one is yes. The difficulty, all aging diseases are multifunctional. I mean, if there's evolutionary reasons for that. I think if you could demonstrate that an age breaker genuinely broke ages and generally broke them at a reasonably high rate, the cross length, then you could ask, in what tissues does that do something significant, and does it do something in the aorta? Does it open up the structure sufficiently so the endogenous clearing mechanisms can remove some of that calcium, can remove the accumulated lipid, and so on? Or is that not the bottleneck? Do you actually need to find some way of getting rid of the calcium, getting rid of the transglutaminase cross-links? I mean, we don't know. And that's why I sort of go on to if you want to find out, you really have to sort of probe it with something that changes the system. It may be we invest 20 years and so many $1 billion getting a drug into phase 2 that's an absolutely authentic, 95% efficient glucosapane cleaving agent. We put it into man and absolutely nothing happens. Then we'll say, oh poo, and go on to the next one.